Welcome everyone to the webinar. For today's session, we have Olivia Angelin-Bonnet presenting Modeling Gene Regulatory Networks via High-Performance Computing. Olivia is a PhD student at Massey University and currently works on unraveling genotype to phenotype relationships from multiomics data using causality inference methods. Really, really cool stuff. We're so pleased she is here sharing her insights with us today. And between slides, I might admit a few people, so apologies for the pause. But housekeeping, before I pass things over to Olivia, I have a few housekeeping items to cover. So if you're not asking a question or speaking during the webinar, we ask that you keep your audio on mute. But if you do have a question at any point in time, we have a question doc prepared for you guys, which I'm going to post that in the chat if I can find it. And here you go. There is the question doc. Record your questions in there. We might get to them during the webinar and answer them if there's a pause between slides or something in Olivia's presentation. Otherwise, we'll cover those at the end of the session. Lastly, the webinar recording, the webinar is being recorded and will be available on the NeSI website in the next few days. So those of you who registered for this webinar will get an email when that content has been uploaded. And I encourage you guys to keep your video on in the session so it feels a little bit more lively. But if you don't want to appear in the recording, please definitely put your turn your video off by clicking that video icon on the bottom left of your screen. So with that out of the way, I would like to admit a few people and then pass things over to Olivia. Welcome, Olivia. Thanks for being here.
Thank you very much for the introduction. Okay, I think you can see my slides. Yeah, shall I get started?
Yeah, get started, Olivia. Thanks.
Thank you. So thank you everyone for coming and joining today. As Megan said, I'm Olivia. I'm a PhD student at Massey University in biostatistics. For my PhD project, I'm interested in investigating genotype and phenotype interactions using computational statistics. What it means is that I'm interested in the flow of information from the DNA through the expression of genes, which I will define in a moment, and especially through the interaction of molecules at different levels in the and scale in the cells to lead to phenotypic traits in organisms. So phenotypic traits are characteristics of organisms. So for example, if you think of a plant, that could be its height or the color of its flowers. So I really want to understand how the information flows from the DNA, and especially through these complex regulatory interactions to lead to observable characteristics. So first, before I get to the simulation part and the HPC part of the talk, I just want to define briefly some of the biological terms that I will use so that everyone is on the same page for that. So first, I mentioned gene and gene expression. So throughout the DNA, what I call genes are portions of the DNA that contain some important information. So a gene is a portion of the DNA, and this information is used via the expression of the gene. So to start, the gene will be transcribed into messenger RNAs that are basically a free-floating form of this information, a free-floating copy of this information. Then later, these messenger RNAs will be translated into proteins, and the proteins are the actors in the cells. They can perform a variety of functions. So for example, they could be used as messengers, they could be used for the structure of the cell, they could be used as enzymes to catalyze some chemical reactions, etc. So I will refer to these messenger RNAs and proteins as the products of the gene. So the gene products are the RNAs and the proteins. And contrarily to the DNA that stays in the cell, these products have a finite lifetime. So they will be degraded at some point, either naturally or through regulated process. So that's what I'm talking about when I'm saying gene expression. And this is at the level of one gene. But now if you zoom out and you look at the level of the genome, you will find that the expression of genes is not independent from other genes. And on the contrary, the gene expression is a highly regulated process, and that depends on other genes expression as well. For example, one way for the cell to regulate the expression of the genes would be to use other genes' product. For example, think about gene A that will be used to produce protein A. Then this protein A could be used to regulate the transcription of a target gene or the expression of a target gene. So that's all happening in cells as complex biochemical reactions. And we as scientists have found a way to summarize our knowledge about these regulatory interactions in the form of mathematical objects that are graphs that we call gene regulatory networks. So in these networks, the nodes, so the genes, the small dots that you can see in blue and in green are genes. And you will find an arrow from one gene to another or from one node to another when we have some type of evidence that the product of a gene will impact the expression of another gene. So these regulatory networks are very important because they summarize our knowledge about how the expression of the genes are regulated within a cell. And one important question in biology is to understand and reconstruct these regulatory networks. And that can be used to understand the behavior of the cell, or more specifically to answer specific questions like the development of a disease or trying to find cures, etc. So what I'm interested in is reconstructing these regulatory networks. Now, one of the ways to do so is to find a way to measure the expression of genes. So we will do an experiment. Let's say that we are interested in looking at a certain plant. We will take samples from different plants, and then we will measure the expression of the genes in the plants across these different samples. Now, when I say measuring the expression of the gene, it could be, and it is often the case, that we measure the level of RNAs of the genes, but we could also look at the proteins, for example. So any type of measure of the expression of the genes. Now, once we have this data set, we can apply statistical methods that I will refer to as network inference methods to try and understand the regulations between the genes. So a network inference method is actually a very broad term. And under that, you can use different statistical methods to reconstruct these regulatory networks. For example, one simple way to do that is to look at the correlation between the expression of the genes across all the samples. You could also use the concept of mutual information. And the one I'm specifically interested in for my thesis is causality, so reconstructing causal relationship between genes in this data set. So you take your data set, you apply the network inference method of your choice, and the result that you get is a reconstructed regulatory network, in which each node here would correspond to one of the genes in your data set, and then you would put an arrow between two genes if you have some type of evidence in your data set that there is a link between the two. In the term of in causality terms, that could be that we have evidence that the expression of one gene has a causal impact on the expression of another gene. Okay, so I just mentioned that we want to reconstruct regulatory networks from data sets. So why am I talking about simulations? The thing is, when you take an experimental data set, so you had your plants, you cultivated them, measured the expression of their genes, now you apply the network inference method to these data sets. Well, what can you do with the result? It's very complicated and time-consuming and expensive to verify that each link that you find, each interaction that you find exists, and even worse, that each interaction that you didn't find indeed doesn't exist. So we are limited by the fact that we don't know what's truly happening. So the result that we get, we don't really have a way to assess whether they are good or not. The other thing is in the data, so you did your experiment and it involves a lot of steps, and each of these steps can bring some noise, add some noise to the data. So the resulting data set is a bit noisy. And as well, because you're analyzing a biological system that we don't know everything about, it's possible that there is some unknown mechanism that will impact your data in a way that you can't predict. So there would be, in the end, in your data set, unknown confounders that you can't predict their effect on the inference. On the other hand, if you decide to do some simulation, what you can do is decide on a regulatory network of your choice, then use a model of gene expression to simulate the expression of the genes according to this regulatory network. And then you get a data set that looks like what we have from an experiment. You can apply your network inference method. And the importance of doing that is then you have your reconstructed network that you can compare to the true network that you use to generate the data. So in doing so, you have a way to say how well your method performed. You can even go further and decide and look at what type of information your method systematically missed, or was it able to detect some type of relationship, etc. So you have a way to assess the performance of this network inference method. The other thing is, because you're using a mathematical model, you control every aspect of it. You can decide that your model has no biological noise. You can decide to add some experimental noise in your model, simulate again the data. You can control every aspect, and that allows you also to test different scenarios. So let's say I have my network, I use a certain model to do the simulation, and then I decide to change a bit my model, and I can compare the result of the simulation and also the result of the inference reconstruction between these two scenarios. So that's why simulations are very powerful tools, especially in this case of network inference reconstruction. So to go back to our project, specifically for this part, one part of my thesis was to understand the impact of post-transcriptional regulation on causal inference performance. So as I was saying, the statistical methods that we are interested in are the causal inference methods. And the thing is, this is again the little drawing of the main steps of gene expression. People, when they're looking at regulation of gene expression, often focus on regulation of this step, which is the regulation of transcription of the genes. And it is indeed a very important part of the gene regulatory networks. But what I was interested in is what happens when regulation occurs at other steps of this expression process. And that's something that we have found is very important as well in biological systems. So I wanted to have a way of assessing how the causal inference methods will be able to reconstruct network if we have post-transcriptional, so regulation that occurs after the transcription step. So there exist simulators that model the expression of genes when you have regulation of transcription, but we decided to construct a model and a simulator that also include these post-transcriptional regulation steps. That is why we decided to develop the Sysmon R package. So it's an R package that is, if you're interested, available on the CRAN and also on GitHub, and for which I've written a tutorial. If you're interested in trying out, there is a tutorial on GitHub to help you go through the steps. And so if you are interested, the links for these will be on the documents that Megan shared. So for this slide, I just want to quickly explain the different steps of the Sysmon R pipeline. And it all starts with the user input. So the user will have complete control over the type of regulatory network that they want to generate. So let's say how many genes you want in your system, the ratio of protein coding versus non-coding genes, different parameters of the regulation, so how much regulation, how many regulators, etc. So Sysmon R takes this input and we'll use it to generate a list of genes. So when I say gene, I say, okay, we want 20 genes. Then we will create an entity. Each gene will have some kinetic characteristics. For example, the rate at which the gene is transcribed, the lifetime of its RNAs and proteins, etc. So Sysmon R creates this list of genes and as well creates the regulatory network that defines the relationship between the genes, the regulatory relationship among the genes. The next step is then to create in silico individuals. And so when I say individuals, in your head, you can picture little plants if you want. And this is because if you remember, I was saying that for reconstructing regulatory networks, you want to have the expression of the genes across different samples. Especially for causality inference, it's important that we have first a lot of samples, from which we can draw observations. And as well, it's important that the samples have some small variations between them. And small variations here mean genetic mutations. So if you think about a sample of humans, we all have unique mutations between us, and that's the same thing for any organism. And these unique mutations will impact very slightly the regulatory networks, and that's what's going to help us to reconstruct the network using causal inference. So in our case, each of the in silico individual will have the same list of genes and the same regulatory network, but they will also carry genetic mutation, meaning they will have actually slightly different versions of the different genes. So all the individual will have the same gene 1, but maybe one of the individual will have a slightly different version of gene 1, for which the RNAs are have maybe a smaller lifetime or something like that, or the transcription rate could be a bit reduced, etc. So that's what I mean when I'm talking about individuals. And in here, individuals refer to these different samples in our expression dataset. Once you have your genes and you have your network and you have your individual, it's time for Sysmon R to create the stochastic model that will be used for the simulation. A stochastic model is composed of two things. The first thing is a list of all the molecules that exist in the system. So here, let's imagine that we're focusing on gene 1. Gene 1, what is it actually? Well, it's first a portion of DNA. So we're going to create a portion of DNA that we call DNA 1. There will also be some RNAs for gene 1, so we'll call them RNA 1. Same for the protein, protein 1. So we build like that the list of all molecules that can exist in the system. And the second thing is the list of biochemical reactions that can occur in the system. So again, focusing on gene 1, we know that the gene needs to be transcribed. So we start from a molecule of DNA, DNA 1, that turns into the molecule of DNA plus the RNA. That's the transcription step. Same thing for the translation. We start from a molecule of the RNA of gene 1, and we produce a protein of gene 1. So that's the easy expression step. And then you have more complicated biochemical reactions that relate to the regulation of one gene's expression according to another gene product. Once Sysmon R has this stochastic model, it can then use it to simulate the expression of the genes over time. So the behavior and evolution of the system over time. And to do that, it will simulate every firing or every occurrence of the biochemical reactions in the system to arrive to, for each gene and each individual, the expression over time, the abundance, if you want, over time of the RNAs and proteins of the gene. So that's what's the outputs, what I refer to as the simulated gene expression. Okay? So actually this step of simulating the expression of the genes is the most computationally intensive step. So when I was developing the package, I figured that actually R might not be the best way to do that. So the reason why it's it can get very computationally expensive is that it's okay to generate every occurrence of some reactions if you don't have a lot of species in your system or if you don't have a lot of reactions. But as soon as you get a large system, so a lot of genes, a lot of regulations, the number of species increases, the number of reactions increases, and then it gets very complicated, not complicated, but very intensive to simulate all the reactions in the system. So that's why R might not be the best way. We know that there exist some languages that can be faster than R for some tasks. So that's why I decided to have a look around beside R. So to go from the stochastic model to the simulation, I use an algorithm called the stochastic simulation algorithm. And actually, it turns out that it has been implemented in different languages. So there is an implementation as an R package. There exists R package that use C++ code to do the actual simulation. And there's also an implementation in Julia. So what I decided to do is to take a system, the same system, and simulate it with the 3 different implementations. And then to try and increase the size of the system, so the number of genes that I simulate, as well as increasing the time for which I want to simulate the expression of the genes for. And for each simulation, each run, I looked at recorded the running time of the simulation. So what you can see here is that in blue, you have the R implementation. And as soon as you get a system that is quite large, or you want to simulate the expression of the genes for a long simulated time, then R can get quite slow compared to the other implementations. So that's what I was imagining, that R would maybe not be the most performant implementation. On the contrary, if you look at C++ and Julia, for large systems, they're almost equivalent. Now, one question that you could have is then, why is Julia so slow when you have very small system? And that is because of the way I use Julia. So I wanted to have an R package that people could use when knowing only R. So actually, that's what I'm going to present next. But briefly, I'm using R, and then I'm sending from R the instruction to a Julia process, then running the simulation and taking them back. So when you have a small simulation that runs really quick, actually most of the time is spent in sending the data back and forth. And that's why you have quite a slow start for the Julia implementation. So yeah, I figured that maybe R is not the best way to go, but I could use Julia or C++, and Julia sounded like fun. So I decided to go for using Julia for the actual simulations. And I did that by using a package, R package called XRJulia. So now imagine you're Sysmon R user. You're starting your R session, you're loading Sysmon R in your library, and then Sysmon R will call the XRJulia package to create a Julia process on your computer with a socket connection between the two, so that each side can send information and then listen or get information from the other side. So then you start to create a network. In the R session, Sysmon R will create the model, send it to Julia. Julia will run the simulation faster than what you could have done with R, and then send them back. So from the point of view of the user, you only have to deal with the R session. You don't have to deal with Julia. You don't have to know anything about Julia programming to be able to use Sysmon R, because everything is done internally via the XRJulia package. So yeah, that's how Sysmon R works. And now the last part is, okay, but why am I talking about HPC computers? So before we move to this section, and I explain why I needed the NeSI HPC platform to run actually the simulations for my project, just to show you physically where my simulations were running. This is the Macwica HPC right there, and that's the one that I worked on. So coming back to our project, if you remember, it's about understanding how post-transcriptional regulation affects causal inference performance, methods performance. I wanted actually to test a few different scenarios. So imagine in the regulatory network, there's only a regulation of transcription. Now, imagine that you actually have a lot of post-transcriptional regulation. Imagine you have a bit less, etc. So I actually wanted to test the performance of the methods for 13 different simulation scenarios. And also because I want results to make sense and to be kind of reproducible, I don't want to simulate one network per scenario, but I want to have a bit more than that. So I decided to simulate 20 networks for each of my scenarios, which leads me to already 260 networks, which is quite a lot. But now, because I don't want to repeat the simulation one time, but I actually want different observations or different individuals, and I want quite a few of them to ensure that my causal inference method has some data to work with, I decided to simulate 3,000 individuals for each of my networks, which leads me to 780,000 simulations to run. Each of them taking, it's a very rough estimate, but taking around from 4 to 17 seconds to run. So it might not seem like a lot, but if you run them one after the other, I estimated, and again, it's a very rough estimate, but it would take around 50 days for my computer, my laptop, to run all the simulations one after the other. So I couldn't possibly stay in front of my computer and look at it for 50 days waiting for the data. So that's why I had to find some way to scale up my work and to be able to run that in a more efficient way. And that's where the NeSI HPC platform comes into play. Because one of the amazing things that they have on the Macwica HPC platform is they have a lot of cores. Now, if you think about the simulations, I have 260 networks, and each of them are independent from one another. So actually, instead of running the 700, etc., thousand simulations one after the other, I could simply run my 3,000 simulation per network independently. And so that's why I say that I can parallelize my simulation. I can run them independently from one another. So what I could do with the Macwica HPC platform was to send each network on a different core and run actually the simulation on different cores. The other thing that made my use of the NeSI HPC platform very enjoyable and very useful for this project was, first of all, R and Julia were already installed on there. So if you're a bioinformatician, you know the struggle of, okay, I need to install this package, but oh no, it's not the correct version, etc., etc. Here, I didn't have this problem because not only R and Julia are already there, also there are different versions from which you can choose from. And even more than that, there's also a lot of documentation on how to install the R or Julia packages that you need. So it was really easy to go from a local computer where I have the versions that I need, etc., to the Macwica HPC platform. The other thing is that, okay, I will generate my networks on my computer so that I can have a look at them, etc. And then I need to send them to the platform and then run the simulation and take back all the results so I can do the analysis on my computer. And the really easy way to transfer the data was by using Globus. So basically, if you use it, it looks like that. You have the two sides of your transfer endpoints, where one of them would be your NeSI session, kind of, the files where you have your simulations. The other side would be my computer. And then I can just simply transfer the data from one side to the other. And it's very easy and fast, even though some of the data were really large. So that was, again, very easy to use. Another thing was the NeSI research support team. That was really amazing. So as soon as I encountered any problem that I couldn't solve, I can just send a ticket, and they're very fast and very helpful. So I won't have to struggle by myself to try and understand why is it not working or something like that. They will be really ready to help me. So that was experience. And the last thing is it's very easy to use, and I'm going to illustrate that in my next slide. So if you're doing any kind of simulation work, ideally, you want to make sure that what you want to do works beforehand. So for my project, I first, on my own computer, developed the R code for the simulations on a very small subset of networks and a very small number of simulations, just to make sure that the code actually does what I want it to do and that I record the data, etc. Once I have that, putting it on the Macwica HPC platform and running that was a breeze. The only thing I had to do was write a Slurm script to execute my R code on each of the cores. And it was as easy as writing 3 lines, basically. I don't have to learn an entire other language. I don't have to rewrite my code to make it work, etc. It was really easy to go from my laptop to the HPC platform. The one thing I had to do is that when you're running these simulations of this scale, you need to ask for how much time you want it to run for and how much memory you need. And if you ask for too much memory or too much time, you're kind of wasting all this time that is actually not used. If you ask for not enough time, for example, then after the allocated time, your simulations will be shut down because it's exceeding the time you asked for. So that would be a problem, right? So the first thing that I had to do before running my entire simulations was to do a bit of scaling to understand the resources that I would need for my whole simulation. So the thing is, for each of my network, I have to repeat the simulation 3,000 times, which are the number of individuals. And these simulations are very similar to one another. So actually, the time that it takes to run one would be approximately the same as the time it takes to run another one. And it's not comparing different networks, it's just inside one network. So what I did actually was for each of the networks, I started by simulating a very small number of individuals, 10. Then I measure the time that it takes to run, the memory that it takes. Then I run a bit more. Again, I record the resources that I use. And I could use that. I had the chance that it was very linear with the number of individuals that I simulated. So using this very small subset of simulations, I was able to generate an estimate of the running time and the memory, total memory needed for my 3,000 simulated individuals. Once I have that, I can go into the Slurm script and say, okay, I need that much time, that much memory, and I can just send the execution of the simulations and wait for an email telling me that it's done. So that was really easy to go from my local computer to the HPC platform. Now I'm still analyzing the results for this project, but in the meantime, for the seminar, Dini did a whole lot of simulations for one small network. So I just want to share why it's really important to be able to scale up your simulations and not just work on a local computer, but be able to run a whole lot of simulations. So for your information, the small system that we simulated, that Dini simulated, has 20 genes. Some of them are regulated. So this is the network of the regulation. And one of the things that can help you understand why it's important to do the simulation is if we focus on one of the gene, which is gene 3, and we look at the initial abundance of the RNAs at the bottom and the proteins at the top for this gene across all the individuals that were simulated, so 6,600. You can see that the initial abundance is not a single value, but it's actually a distribution, which looks like what we would see in experimental data sets. And in this distribution, you have the play of biological noise and also of genetic mutations, which we wouldn't see if we were only to make a few simulations, because we would not have a lot of variation. And as well, because we have so many simulations, we can actually zoom in and check that actually when individuals have different versions of the gene, so the different versions I refer to as allele. And so when we have different allele, when individuals, have different alleles of this gene 3, they actually have a bit different values of initial abundance for their RNAs and proteins. And again, so so for all individuals carrying one same allele, you see a bit of variation, which is the biological noise. But then between different versions of the gene, you see as well quite a bit of variation, which corresponds to the genetic variation between individuals. And the other thing is you can go from the left side, which is looking at the expression of a gene for one individual. So this time we're looking at gene 17, and we're looking at the evolution over time of their RNA and their protein for one individual. You can go from there, where you can see a bit of variation, to looking at an entire population. So looking at 100 individuals and then looking at, okay, there's some biological noise, but there's as well difference in the behavior due to genetic mutations. So that's quite interesting to be able to go from a few simulations, because that's all your computer can manage, to looking at a lot of simulations. So in conclusion, there's two things that hopefully you will remember from this talk. Is first that simulations are awesome, and you don't have to have experimental data sets to do good science. You can work on simulations as well. There's a very insightful way of doing some research. For example, if you're interested in a particular biological system, you can use the knowledge that you already have about the system to build a model. And this model can help you by doing simulations, etc., to test hypotheses. So you can simulate it, realize that the data does not quite look right. Okay, maybe there's something we're missing. There's actually a mechanism that we don't know. You can change your model, do comparison between different versions to test different scenarios. And again, you have this aspect that you can control the complexity of the data. So in the case of testing some statistical method performance, you can start with a very simple model and try to increase the complexity and see how your statistical methods will react to that, how well the inference will go. And the second thing is, if you're already doing simulation work, or as well, if maybe I convince you to try, the NeSI HPC platform is really ideal to scale up your work. And as I was saying, there's a lot of help, there's a lot of resources available. And so I found it really easy to go from working on my local computer to scaling up my work on this HPC platform. So before I end this talk, I would like to thank Massey University and Plant and Food, and especially my supervisors, Mathieu, Patrick, Suzanne, and Samantha. And I would like also to thank NeSI for letting me talk a bit about my research, and especially Dini and Megan for helping me for the seminar and running the simulations, etc. So thank you very much for you as well for listening. And if you have questions, please don't hesitate.
Thanks so much, Olivia. That was fabulous. I just have to say every time that you're such a great presenter and you can communicate your research so well and so clearly. So thank you for that. Really glad to have you. There are a couple of questions on the question doc. I'll go through those, but just a reminder to everyone in the room, if you have anything else that has come to mind, you can keep popping those in the document. We're just going to go through those for as long as we have until we finish them. So first one, how do you tag the nature of interactions, network edges, when constructing your network? For example, in your network, what would be the difference between up-down regulation?
That's a very good question. So when I'm building the model in R, it's all data frames. So a regulatory network would actually be a data frame where each row corresponds to one interaction. And so in a data frame, you can use different variables to give specific details about the regulation. So for example, I would have one column that tells me whether the regulation is actually positive, activating, so the regulator will increase the expression of the target gene, or whether it's negative, so the regulator will actually decrease the expression of the gene. I also have a column or a variable that tells me which type of regulation it is. And then when it comes to visualization, then I use different colors for the user to be able to quickly see the difference. I also have options for the user to only look at regulations that are of a specific type, and I use as well line types, so the dash, etc.
All right. And next question, what parameters are used to simulate a regulatory network given the real data is probably too noisy? How many well-curated networks Sysmon R uses to estimate parameters of the regulatory model?
Oh yeah, very good question as well. That's something that we've put a lot of thought into, because one of the things in simulators that I found is sometimes the user is left to give parameters, but actually if you don't know what's happening in biological systems, you can use parameters that are quite far away from the truth. So what we did is we looked at in the literature experiments that focused on estimating, for example, the lifetime of RNAs for some model organisms. So the thing is, there's a lot of data about yeast, so I focused all my default values in Sysmon R are from experiments in the yeast, if I could find them. And so there are experiments that focus on what's the distribution of observable lifetimes of RNAs in yeast. And so I use that to derive the values of the parameters, plausible values of the parameters. And then I did some simulation and compared that actually what I got as a result was similar to the data given by the experiments.
Fabulous. Okay, one more question at the moment, unless anyone else wants to add things to the document. But the third question, might biosynthetic gene clusters, genotype, and the molecules they produce, phenotype, be a possible initial experimental test case as opposed to something as complex, relatively speaking, as, for instance, a plant?
Yeah, that could be. So actually what we've done, one of the small networks that we simulated as a proof of concept was a small network of endothymine biosynthesis regulation in plants. So it's a very small, only a couple of genes. And then we checked that what we got out of the simulation looked like, even though it's not completely equal, because again, there's some mechanism that we missed, but it looked a bit like experiments that were done. So at this stage, we don't, in Sysmon R, we don't have the phenotype part yet, so we only have gene expression. But of course, simple networks from which we have some knowledge and we know what we should expect are a good example of testing Sysmon R to check that the model makes sense.
Great, thanks for that. That's all the questions. Manuel, is that a hand? Sorry. No. Okay. I think that's all the questions we have for the moment. And I would like to finish off by just showing a few other bits of information where you can get some more information. If you do have questions that come up while I'm doing that, feel free to pop them in the document because we do have a few more minutes. But let me just share my screen. So before you go, first of all, thanking Olivia again for that awesome presentation. Such a great communicator. Loved hearing about your research. And I'd just like to point out a few tidbits where you can get some more information. If you haven't noticed, there is a resources section at the top of the question document that we have been using today. So if you want to check out more about the Sysmon R package, more information on Globus, or how you get answers to your NeSI specific questions, there's some information there for you. If you do have NeSI specific questions, as always, please email us at support@nesi.org.nz. We are always happy to hear from you. And last but not least, the webinar recording and slides will be posted soon, so keep an eye out for that. And do feel free to pass it along to your colleagues if you did find that this was helpful for you today. I see a question that has come up, but I'll just finish my slides real quick. Before you guys go, I did want to highlight a service that NeSI provides that some of you may be interested in. High performance computing is a tool, and most researchers aren't going to be HPC experts, nor should they be. You have enough on your plate already. So that's why NeSI has a team of research software engineers who can help you through a consultancy project. For researchers at most universities and CRIs, this consultancy service is provided at no cost to you, but you do have to reach out to us so that we can chat about your project and what kind of help you're supposed to or hoping to get from us. If that sounds interesting to you, just flick us a message at support@nesi.org.nz. I'll stop sharing my screen and I'll ask that last question. So how easy or hard to do parallelization with Julia for your project? What kind of scheme do you use? For example, MPI?
It was not really complicated in the sense that so I have parallelization if you're running Sysmon R on your own local machine and you want to, for example, simulate different individuals on different cores if you have a lot of cores on your machine. And this I do using the parallel package in R, and I can't quite remember which of the scheme I use, but it's I can check and put it in the document if you want. Then on the HPC platform, it was even easier because literally you can write a script that will R script that will do the simulation for one network, and then on the Slurm script that you use to run the R script, you can say that you want to run this script on that number of cores. And so there is an environment variable that is created with the ID of the core if you want. And then you can use that in your R code to say, okay, I want to run network number 22, etc. So it was very easy actually.
And I think Eli is going to elaborate a bit on his question. So Eli, go ahead.
Olivia, can you guys hear me? Cool. The question I asked about, like, how did you estimate your parameters? I mean, well, I was lucky because I was working in the youth lab, and I know the data that is out there. This is like roughly, but 80% of it doesn't validate in, in lab work. So I mean, like one good way to check if, if your simulation is doing what it should be doing is always good, good to just like compare different organisms with each other and see like you get the same result. But otherwise, I mean, I did try to go quickly through the paper, but I didn't see any kind of test that say, okay, I came up with this model. Now I'm testing it against this good known data set to validate, if what I'm getting is just like is real result or I'm generating more noise. So that probably would be a good thing, to look at. Thanks.
Yeah, no, that's a very good point. And it's actually quite tricky because in the end, there's so many parameters in the regulatory networks. So even if we were to use the best estimates that we have for some of the parameters, there are still some, like what's the binding rate of one specific regulator onto the DNA for the target. There are still these parts that we have to give our best guess and use some data that is available out there, but may not be the best. The thing that you're looking for, because the paper is so small, it's in 2 pages. Everything is in the supplementary. And so I think what you're looking for would be in supplementary number 2, I think, or 3. So I did quick simulation of the plant regulatory network that I was mentioning. And the other thing is Sysmon R simulates absolute abundance, right? And most of the data we have is not absolute, actually, it's relative, you're comparing two things. So I was more looking at the ratio between the RNAs. The other thing I did was, if you simulate a bunch of genes without any regulation, looking at the ratio between RNAs and protein looks like what we've seen. But again, you're completely right. The data sets that I've used could not be the most accurate. It just, we had to make a choice in terms of, there are so many parameters that we want to use in the data, in the model. So go with the model organisms for which we have the most information. But I agree, if you change the parameter, will it give you something that is good for plants as well, or something like that.
Cool. I'll just ask if there's anything else that maybe Denny, did you want to mention anything else? Olivia, any last comments? Are you happy? We got thumbs up. We're happy. Cool. Well, if you guys would unmute yourselves and join me in thanking Olivia. That was a fabulous presentation. And then we'll be done with the webinar. Have a great afternoon, guys. Thank you, Olivia.
Thank you very much.
Thanks, Olivia. Well done.
Thank you.
Well done. Have a great afternoon, guys.